In this video, we are going to use the bacterium, Mycobacterium tuberculosis, to describe how scientists use the germ theory of disease and Koch's postulates to prove that a specific microbe causes a specific disease. Mycobacterium tuberculosis is a rod-shaped pathogenic bacteria that has a unique waxy coating that prevents gram staining, and this coating is due mainly to the presence of mycolic acid. This bacterium causes tuberculosis, a very contagious bacterial disease that primarily attacks the lungs. Robert Koch discovered this bacterium in 1882 and linked the bacterium to the disease. So how did he prove that this specific microbe, Mycobacterium tuberculosis, is the causative agent of tuberculosis? Well, first, we must look at the overarching theory called the germ theory of disease, which was developed mainly by Louis Pasteur and Robert Koch. The theory is that microbes cause infectious diseases. Koch was the first to prove this theory using experimental steps called Koch's postulates that proved that a specific microbe causes a specific disease. Now let's run through an experimental setup to describe how we would apply these postulates to prove that Mycobacterium tuberculosis causes tuberculosis. For ethical reasons, these experimental steps should be done on only an animal model. Some microbes can only infect humans, and animal models cannot be used. But for this video, we will use mice as our model. The first step of the postulates is to ensure that Mycobacterium tuberculosis is present in all cases of the disease and absent from healthy animals. An exception to this is certain microbes can be found in both healthy and sick individuals. To test this first postulate, blood or tissue samples can be taken from both healthy mice and mice with tuberculosis and analyzed under a microscope. Mycobacterium tuberculosis can also be found in healthy patients, but for the purpose of this video, only the sick mice would have Mycobacterium tuberculosis in their samples. The second experimental step or postulate would be to isolate the bacterium from the mice with tuberculosis and grow the bacterium in a pure culture. An exception to this is some microbes are unable to be grown in a pure culture. Once a pure culture of Mycobacterium tuberculosis has been derived, a healthy mouse can be inoculated with the pure culture. The third postulate is to observe the pure culture causing tuberculosis in the healthy mouse. The final step or postulate in order to prove that the bacterium causes tuberculosis is to take a sample of the inoculated mouse and isolate the bacterium into a pure culture again. Now that we have satisfied all four Koch's postulates, we can conclude that the Mycobacterium tuberculosis is the causative agent of tuberculosis, and this confirms the germ theory of disease, which is the theory that microbes cause disease.